everyone, welcome back to this video related to chapter 6.2 of CBSE Biotechnology course, which is plant cell culture and its technologies. In the previous two videos we have covered introduction to plant biotechnology, plant cell cultures, and in the second video we covered the methods involved in plant cell culture and some of its application. In this video we will cover how we make transgenic plants and other methods and the methods related to gene transfers in plant. So before we discuss methods related to gene transfer in plants, we need to understand a little bit why and how we need these technologies and what are the limitations for conventional breeding. I'm sure by now you know what do we how we actually do conventional breeding. So normally in conventional breeding we identify two contrasting cultivars of same species most of the time, and we try the contrasting cultivars basically differs on the traits of choice. For example, disease resistance or abiotic stress resistance or increased yield. When we cross them, then most of the time it has been found that the beneficial effects they are more visible in the hybrids which are made by the crossing of two parents. However, this has certain limitations, some of which are listed here. That conventional breeding is mostly done in the same species or the species which are very similar. In other words, this method would not work between two plants which are basically from two distant species, or in any case if they are from different genus of plant kingdom. Second is the limitation in conventional breeding is dependent on the targeting of the traits. When we basically do the crossing or the sexual reproduction of two individuals, then it is basically mixing 50-50% of both the genomes. So 50% comes from male parent, 50% of the genes coming from female parent. So basically it is a untargeted mixing of 50-50% traits. So we cannot assume or we cannot guarantee what type of uh, the traits would be shown by the offspring. This is also related to the undesirable gene transfer because it is not targeted. We are just mixing 50-50% genes from both the parents, and the one of the major concern is it takes very long time, because if we are targeting for one single trait by conventional breeding, then after making hybrid by in first generation we need to do selfing for 7 or 8 or 9 generation through backcrossing. I'm sure you know what is backcrossing, and uh, it takes a very long time. Also, we need a very large area to basically make these crosses because, as I said, we cannot expect the desired outcome by one single cross. In general, when the breeding institutes they do breeding for a certain specific crops, hundreds and thousands of crosses are made, and you can imagine then if we are making hundreds and thousands of crosses with different combinations, then we need to grow those resultant plants and screen them in the field. On the other hand, genetic engineers gives us an opportunity by which we can target one single gene from any of the useful source, and this source can be a bacteria or any distant organism where we can identify the work of that gene, and through genetic engineer we can transfer that gene into hypothetically into any plant species where genetic transformation methods are available. So the major uses of genetic engineering technology are listed here. The first and foremost use is the gene switching. As I said, if we identify a gene or a pathway in a certain plant or maybe in a distant organism, say for example bacteria or any other, even animals, we can transfer the gene or those genes from that organism to our target plant species. Genetic engineering methods are also being used for other things such as plant disease diagnostics, and making crops which are good for biofuels, making plant and tree species which are more tolerant for degraded ecosystems so that they can be used for bioremediation. Plants and vegetables, plants mainly are being tested, and the technology are being developed so that they can be used for molecular farming and production of biopharmaceuticals and nutraceuticals. And the very important, and we will discuss in detail also, genetic engineering for food crops for enhanced biofortification and enhanced value-added products in target food crops. We will discuss some of these examples in coming slides. So what are the major steps in plant genetic engineering? There are 5-6 major steps by which we basically achieve plant genetic engineering. And the first thing is the identification of a target gene or genes related to a target pathway. So by gene, I mean a single gene if it is of our use or if it is providing the required function. And pathway means there are more than one gene sometimes which basically carried out a complete pathway for formation of a desired product. So after identification of that gene, DNAs are extracted and cloning of that gene is done. We study the mechanism of that gene action, and then we try to modify the expression of that gene by swapping or by changing the promoter activity. After that, when it is transferred to the plant, the safety aspect of gene and its products are done. We will discuss what do we mean by safety aspects and what are ethical issues involved in plant genetic engineering. So there are many methods available for gene transformation in plants. They are listed in this table. Uh, Important of them are the TI plasmid mediated gene transfer formation, where we use a bacteria, Agrobacterium tumefaciens. We will discuss what is that method and what is special about this bacteria. Another is physical gene delivery by using microprojectile bombardment, or also popularly known as gene gun method. And then there are other methods which are viral vectors, direct gene transfer into plant protoplast, microinjection, electroporation, and liposome fusions. So microinjection, electroporation, and liposome fusion are again mostly used by isolated protoplast only. I'm sure you remember from the last slide what do we mean and how we do plant protoplast isolation. And even if we are not doing genetic engineering, plant protoplasts are basically useful for many other methods, such as production of somatic hybrids and hybrids. So, Agrobacterium tumefaciens, also known as nature's own genetic engineering. So, it has been known for many years now that Agrobacterium is a bacteria, soil bacteria, gram-negative soil bacteria. I'm sure you know what do you mean by gram-negative and gram-positive staining. And it causes a disease in plants which is called crown gall disease, as you can see here. The region above the soil where various branches proliferate, it is called the crown region because it looks like a crown. And the gall is an unorganized mass of cells which is most of the time is near the crown region or on the crown region itself. So when agrobacterium infects a plant, it causes this disease called crown gall disease. Although we call it a disease, but apparently there is no harm to plants apart from the appearance. It looks bad. And how agrobacterium does it, we will see in the next few slides. The thing is that agrobacterium normally stays in soil inactive, and if a plant gets wounded by some physical or mechanical things, then the plant cells which are basically got wounded and trying to recover, they release protective or I would say useful chemicals which are mostly phenolics in nature. And that phenolic compounds, when they are released in soil, they attract agrobacterium towards the wounding site, and then agrobacterium basically attaches itself to the cells which are damaged by wound, and then the transfer of its some part of its own genome happens. So the part which from which the transfer happens is known as TI plasmid. So TI is tumor inducing plasmid. TI stands for tumor inducing. So it's a plasmid inside agrobacterium. So I'm sure by now that you know what is a plasmid. Plasmid is an extra chromosomal DNA which is found in bacteria. It has its independent DNA replication machinery, and when bacteria divides, then this plasmid also gets replicated, and they are also incorporated into dividing and progeny bacterial cells. So coming back to TI plasmid is fairly a very large plasmid, 200 kb or around 200 kb in size, and there are various regions which are there on this TI plasmid, and most important of that region is tDNA. tDNA stands for transfer DNA. So this is basically the DNA which is transformed from bacterial cell to plant cell, and then this DNA, by a mechanism which is governed by again TI plasmid based genes, it is incorporated into plant genome, and then afterwards these genes start making products inside the plant cell which are not useful for plant, but then which are useful for bacteria for its own use. So apart from tDNA, other important regions are Vir genes and opine catabolisms. And as I said, that the replication is independent in bacterial cells. So the TI plasmid also has an origin of replication from where the replication of this entire plasmid starts. So if we see the important features of transfer DNA, there are two inverted repeats which flanks transfer DNA called left border and right border. And this left border, right border has genes for auxin biosynthesis, cytokine biosynthesis, and special amino acid synthesis. These amino acids are collectively called opines. I'm sure you remember what do we mean by auxin and cytokine. These are plant hormones, and if we provide in these hormones in excess amount, rapid cell division starts. Even in plant tissue culture, if we provide high amount of auxin and cytokine in the growth media, then our cell starts rapid division, cell division, and our explant makes callus. I'm sure you remember this thing from video number 2, or you can still refer to video number 2 for the effect of auxin and cytokine on plant cells. So same thing happens when tDNA is transferred to plant cell. In tDNA there are auxin biosynthetic genes and cytokine biosynthetic genes, and when these genes are incorporated into plant genome, these genes start showing their effect. Those cells where tDNA is transferred, they start making higher amount of auxin and cytokinin, and when higher amount of auxin and cytokinin is there, then those genes basically starts dividing uncontrollably, and that's why they make this cancer-like or gall like structure, an unorganized mass of cell called crown gall. Another thing which is made by the effect incorporation of tDNA into plant genome is special amino acids. I said these are collectively called opines. So opines are unusual amino acids. These amino acids cannot be used by plant cells for its use. On the other hand, agrobacterium use opines for the source of food material, for example carbohydrate, for example nitrogen, and other important compounds. So what happens if you understand by this, that if plants wounding happens, agrobacterium by sensing phenolics, agrobacterium attaches itself to the wounding part, and then transfer of tDNA happens from TI plasmid of agrobacterium to plant cell, and this tDNA has useful genes for bacterium, not for the plant. So these genes start making more of auxins, more of cytokinin, and opines. The collective effect of more auxin and cytokinin is that the target cell, they start dividing, and then they make a cancer-like structure or unorganized mass of cells, which we call crown gall, which is visible near the crown region of plants, and this disease is called crown gall disease. So soon scientists realized that anything which can be put between these inverted repeats to 25 base pair in around 25 base pair inverted repeats called left border and right border, if we remove everything from here and we put our gene of interest, that can be transferred into plant cell, and that basically was a very lucrative or very attractive opportunity. So the two basic things which hampers initially use of agrobacterium for plant genetic engineering is that TI plasmid is very large, and we cannot maintain a TI bacteria having a plasmid which is 200 kb under normal conditions in our laboratories. Second is the tDNA of TI plasmid has these tumor inducing properties, increased amount of oxygen, cytokinin, and formation of pines, which are basically not useful for genetic engineering purpose if we want to do targeted genetic engineering related to some trait improvement. So these two problems were overcome by making what we call a binary vector system of disarmed TI plasmid. So what scientists did in laboratory, first, the agrobacterium itself is kind of genetically engineered, but for using agrobacterium in genetic engineering experiments. So genes from TI plasmid, genes related to oxygen synthesis, cytokine synthesis, and opine synthesis, they are removed. Only left border and right border is retained in the modified tDNA. Also, there is another region which called virulence region, and which has virulence gene, or also called Vir genes. They are removed from TI plasmid and put it onto the main genome of agrobacterium bacteria. There is a large section or large part of TI plasmid which is basically having genes by which agrobacterium use or catabolize opine, and since we are not interested in biosynthesis of or synthesis of opines, we don't want opine catabolism genes as well, and they are also removed. What scientists also did, that there is already an origin of replication for agrobacterium. They introduced one more origin of replication, which is used for replication of this disarmed plasmid in another bacteria, popular bacteria called E. coli. So this is called binary vector system, and the plasmid is now disarmed TI plasmid. So the resultant plasmid looks like this. It has two origin of replication, both for agrobacterium as well as E. coli, so that it can be maintained in both E. coli as well as in agrobacterium. Left border and right border are there. Target gene is inserted between left border and right border, and for basically measurement of transformation efficiency and to screen out the transformed cells from untransformed cells, a selectable marker gene is also introduced between left border and right border. What is selectable marker gene, we will discuss in probably in the next slide. This binary vector or modified TI plasmid is then inserted back into the agrobacterium, and then plant tissues are used for to basically infected by these modified agrobacterium cells, and we get transgenic plants. So as I said, what are the selection marker genes? So basically, if we do infect a group of cells, not all cells in a particular tissue or in an experiment gets infected by agrobacterium, only some of them gets infection and transfer of this modified tDNA. But how to differentiate between a transformed cell and untransformed cell so that we can grow only the transformed cells? In tDNA, selection or reporter marker genes are also inserted. Selection marker genes are mostly an antibiotic or herbicide related gene. When we provide these chemicals in the nutrient media, then only the cells which got transformed by agrobacterium experiment and which has these genes, they survive. Other cells dies. The most common examples of these genes are NPT2, which cause for detoxification of neomycin types or kanamycin type antibiotic, and the full name is neomycin phosphotransferase. Similarly, there is another antibiotic called hygromycin, and the gene which can basically degrade this antibiotic is hygromycin phosphotransferase. Then there are certain reporter genes which basically gives us visible uh, hints which genes are transformed and which cells are transformed and which are not. They are called reporter gene, and common reporter genes are beta glucuronidase synthase gene. It is an enzyme which is used for a histochemical assay, or popularly known as Gus histochemical assay. When this gene is present in a cell, provided and we provide suitable substrate and other condition, those cells turn into blue. So if it is present in a plant cell, then plant cells turns into start showing blue color. There is a herbicide resistant gene also there, which is called bar gene, and it provides resistance towards herbicides. And another interesting reporter gene is green fluorescent protein. It is taken from some sea animals and fish, and if we provide ultraviolet light, then basically these cells start growing into a fluorescent green color if GFP gene is there. So by these method, by a mix of selection marker gene and the reporter marker gene, we basically differentiate between transform cell and non-transform cells. So here I have shown a hypothetical example of a plant transformation experiment. So what we did here, we take anthers of a popular cultivar of wheat, which is good for plant tissue culture purpose. So in one single experiment, we not only made haploid, but we made actually the transgenic haploid. So what we did anther culture, which makes these somatic embryos. I'm sure you remember production of haploid and anther culture from the last video. So when these somatic embryo derived, somatic embryos are made by anther culture, these somatic embryos are infected by modified agrobacterium, as I discussed in the previous slide, which has a reporter marker gene, Gus gene is there, and also plant selection marker gene, which is kanamycin resistant gene, NPT2. So these, after infection experiment of agrobacterium, these are grown on a media which has kanamycin as a selection agent, and only the somatic embryos which were transformed by agrobacterium, they start growing on, they grow into green plants on uh, nutrient media supplemented with kanamycin. We can also check the gas histochemical assay, and you can see that the somatic embryos, germinating somatic embryos, basically they turn blue by the activity of gas gene, and these are the resultant haploid plants. These similar experiment can be done with other useful explants in any other crop or plant species where agrobacterium mediated transfer methods are available. You can have any other suitable explant depending upon what type of plant we are uh, trying to make transgenic plants. Another method for stable plant transformation is the bialistic method, or as I said, popularly known as gene gun method. So there are two versions of this machine available. One is laboratory uh, um, uh, version, which is used for making stable transformations, and the other one is field method, where we need to make only a group of cells for some time. Say, for example, if we need to make a particular leaf cells for a period of some time, as long as that leaf is alive. So it is basically to test certain biochemicals or certain special uh, agents useful in plant defense and other things. So this is how these machine looks like. The lab version is kept in a laminar flow hood. I'm sure you remember what do we mean by laminar flow hood. It's basically a chamber where sterile air is provided by HEPA filters. So this machine by appearance, it's a pretty simple machine. There is a chamber where we keep our plant tissues, and then there is this hollow tube where we basically put plastic trays which are coated with gold particles having our gene of interest. In this chamber, very high pressure is created by use of a gas. Mostly we use helium gas because helium is an inert gas, and there are no chances of helium of reacting with any of the component which we are using inside the the gene gun, and it is not harmful for plant tissues as well. So we put our plant cells here in this chamber. Gold particles coated with the DNA of target gene is put here, and from above we provide a very high pressure of helium. With that high pressure, basically the gold particle flies with a very high speed, and then they enter into target plant cells by the repairing mechanism of gene repair or DNA repair. Those genes are incorporated into plant gene, and then we basically make those cells regenerate into whole plants, and then we test those plants for targeted effect. For example, if we take these embryoids with somatic embryo, these structures, anther culture derived structure, we can put this structure here in this chamber, and they can be bombarded with the DNA. This DNA will enter into the cells of those somatic embryos, and then these cells, by its own repair mechanism, the genes will be incorporated in the genome, and then the resultant plants would be transgenic. So as I said, these are the two most popular methods of gene transformation in plants, agrobacterium as well as gene gun, direct gene. So they both have their advantages as well as disadvantages. The advantage of agrobacterium method is it's more systemic. It shows less copy number in transgenic, which is actually a desired trait. And when there is a less copy number, then we can assume that there will be higher chances of this transgene expression, and the resultant plant will show better effects or desired effects by expression of the genes which are transformed. On the other hand, the limitation of agrobacterium method is it is only successful in very few plant species, not many plant species are there which can be transformed by agrobacterium. On the other hand, gene gun has a beauty that practically any tissue and any plant type can be transformed, because we need not to we just need to have an explant from which we can regenerate the plant, any type of explant. I'm sure you remember there are so many types of explant available for plant tissue culture depending upon the target plant. Shoot apical meristem, root apical meristem, birds, flowers, leaves, tissues, flower tissues, and many others. So this is the good thing about gene gun. The bad thing about gene gun that gene gun normally results in very high copy number of transgene putting into the cells, and when there is a very high number of genes in a single cell, then it is not desirable, because we cannot many times they do not show optimum expression. They show much higher expression or much higher activity of the target gene, and it is not good for progeny regenerant plants. So other method which I listed in that table is related to DNA delivery through microinjections or electroporation or chemical mediated gene transformation. These are mostly used in case of isolated protoplasts. So in chemical method, the gene cassettes or the genes which are we need to transform into protoplast, it is mixed with a chemical called polyethylene glycol or dextran sulfate, and then in the presence of polyethylene glycol and dextran sulfate, isolated protoplasts sometimes they basically ingest those genes from their surroundings, and then these genes can be incorporated into plant genome again by the same repair mechanism. Microinjection is again it's called of an it's type of an injection only. The needle is very thin, and that needle, through that needle the genes or the DNA is basically injected directly into the protoplast. Electroporation is again a method which is basically used for isolated protoplast only, and in this case protoplast is mixed with the DNA in a liquid media, and a mild temperature shock is provided. What happens with this temperature shock, that this protoplast ingests its surroundings, because during the time of that electric current, the plasma membrane sometimes breaks and it rejoins itself, but during that breakage period, the surrounding solution enters the protoplast. So these three methods actually looks like very simple and pretty straightforward, but the only limitation with these methods to be used is the regeneration from plant protoplast is very difficult. It is very difficult and it is successful only in a very few types of plants where entire plants can be regenerated from protoplast. On the other hand, these methods are very useful in case of studying the effect of a gene or localization of a gene. By localization, I mean intracellular localization. So, for example, if we want to know, we have an important gene and we want to know whether in which part of plant cell it expresses in mitochondria or chloroplast or nucleus or in cytosol. So in that case, that gene is basically uh, can be transformed into protoplast with any of these method, and then we can see where the gene is going through different visualization methods. So far, by using agrobacterium or by using gene gun method, many plant species has been successfully transformed. I list in those species in this table, and you can see many of them are actually food crops. So it is not a very difficult task these days to make transgenic plants by using any of these two methods. And over the period of time, much I mean, uh, agrobacterium mediated transformation methods are developed for many crop plants. So after this, I think we should discuss some important applications as listed in the biotechnology chapter also 6.2. And I would like to start one of the most famous product of plant biotechnology, which is related to production of golden rice. As you can see, this is the normal rice which we use almost on daily basis. It is white in color, and golden rice, as the name suggests, it basically looks golden in color. So as I said, it's basically always a need which results into development of some product. So what is the need for golden rice? So if you see this map, basically it shows the prevalence of provitamin A deficiency in different countries, and you can see in our country is basically clubbed with other countries, mainly in African continent, which has a very severe provitamin A deficiency in its children's and in women. As per some studies, more than 50% of Indian kids below the age of 12 years, they are severely malnourished as far as the availability of provitamin A is or vitamin A is concerned in their diet, and it causes severe growth defects as well as night vision is affected by deficiency of vitamin A. I'm sure you know what are the effects of what is vitamin A. It is retinol, we call it chemically, and deficiency of vitamin A results in poor vision and poor overall growth and development. So in plants, the 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 synthesis of vitamin A or retinol is basically through conversion of beta carotenes, which is precursor for vitamin A. So carotenes are colored compound found mostly in colored vegetables such as carrots, such as capsicums, and tomatoes. These are the ones which are high source of beta carotene. And synthesis of provitamin A or beta carotene is basically a sequential action of certain enzymes which are coded by different genes. So beta carotene synthesis is initiated by geranyl diphosphate, which is converted to phytoin by an enzyme called phytoin synthase. This phytoin is converted to lycopyl by sequential action of two enzymes, phytoin desaturase and epsilon carotene desaturase, and then this lycopene is converted to beta carotene by an enzyme called lycopene beta cyclase, and these four enzymes are coded by four different genes. Our staple food is mostly made up of rice or wheat, and in the seeds of both rice and wheat, these enzymes are not functioning. These are absent. That's why the grains of rice or the grains of wheat, they are white in color, and they are not having substantial amount of beta carotene as the vegetables has, such as carrots and tomatoes. So what scientists basically they hypothesized, if these genes can be genetically introduced into a system where these genes starts expressing in the seeds itself, when the seeds is being made inside the flowers, then we can increase the beta carotene content in the seeds of cereals or in other fruits where beta carotene is not present. By fruit I mean, say, if you see banana is mostly carbohydrate. There is no beta carotene or very less beta carotene in banana. For example, potato again is white in color, mostly pale white in color, and it is again made up of largely carbohydrate, and there is no beta carotene. So what scientists are trying to make, and they have successfully done in case of rice, these the relevant genes are made expressing into the seed itself, and when they are there in the seeds, they basically synthesize, because the precursor product is always present, geranyl diphosphate. It's only the availability of these genes which was required, and then it is mm, the synthesis can be induced in developing seed of rice, or developing fruit of tomato or potato. So this is a hypothetical example what scientists have designed. You can see there is this right border, and it is swept. This is left border and this is right border, and the genes are arranged in a sequential manner. And this tDNA was transformed by the help of agrobacterium into rice calli, and from that rice calli, rice transgenics were raised, and these genes start making beta carotene or carotenes inside rice grain, which looks like this. This is a normal rice which is untransformed, and these two are example of rice which made transgenic. This is golden rice 1 and this is golden rice 2. And golden rice 2, you can see it is basically much yellower, or I would say orangish in color in comparison to normal rice, and this orange color is due to the presence of very high amount of beta carotene inside the seeds itself. So it has been so in many countries the golden rice has been commercially being grown these days, and if a child below 12 years or 8 to 12 years, or mainly the growing age 6 to 12 years, if a child consumes rice as a staple food 2 to 3 times, if it takes a normal diet of around 50-60 grams, or consuming around 150 to 200 grams of golden rice daily, then it can basically have around a large part of daily requirement amount of RDA, what we popularly called as required daily amount RDA of beta carotenes by just consuming rice. If ample amount of beta carotene is provided through the diet, then beta carotene converts into retinol or vitamin A inside the body, and then the problem of vitamin A deficiency is largely addressed. Another famous example and successful famous example of transgenic technology is the production of BT plants. More so, uh, more popular one is the BT cotton. Here you can see a healthy cotton flower, and you can see it is having a good amount of cotton fibers inside. So there are pests. Pests are always there. Mostly farmers, they basically spray pesticides to get rid of different type of pests, and among many types of pests there is this insect pest which is largely called as a lepidopteran class of pest. It's called cotton bollworm. So this is cotton ball, and you see this caterpillar type of thing. This is cotton bollworm, and apart from eating, it thrives on cotton. It basically can eat any part of cotton, but mostly it affects cotton leaves and the cotton balls. So before a cotton ball is formed, it enters inside the developing fruit of cotton, and it finishes everything inside. And when plant matures, basically there is nothing left as far as cotton is concerned, and the farmers they got huge losses. And the conventional method is to spraying or providing plants with by spraying them with insecticides or pesticides. But over the period of time and by indiscriminate use of those pesticides, in most of the world regions, this cotton ball worm become insensitive to most of the types of pesticides, agriculture pesticides which are being used. So no pesticide is there which can 100% or which can effectively control the growth of cotton bollworm. So what people and scientists decide that they basically developed a method where they identify certain genes in a bacteria. Bacteria is Bacillus thuringiensis, which is again, I'm sorry, which is again a soil bacteria, and this bacteria has a group of genes called cryogenes popularly, and these cryogenes basically code for some proteins called delta endotoxins. These delta endotoxins actually have been being used for spraying plants as an insecticide, and if any insect eats a plant part which is sprayed with a delta endotoxin, it dies. So what scientists hypothesize that rather than spraying these delta endotoxins, which basically not an economic method, because we need to grow bacteria and then isolate delta endotoxin from the bacteria and then making it its packets and mixing it with the sprayable granules. Why not make plant to make delta endotoxins inside the plant cell itself, so that delta endotoxin will be present in the plant cell, and if insect tries to eat it, it will die, and the same thing happen. So first they tested this effect of overexpression of those cryogenes in model plants such as tobacco and other model plant, and then the cotton transformation was done by method of choice, and then it was tested against infection of this cotton bollworm, and as soon as cotton bollworm try to eat the cotton which is producing delta endotoxin, transgenic cotton, it dies. Afterwards, the success of Bt cotton was repeated in many other crops, including food crops, where the resistant pest is a problem and no suitable insecticide is available anymore. Most prominent of them is brinjal. Other examples are potatoes, tomato, cauliflower, cabbage, rice, maize, and many other. So when these plants they are transformed with cryogenes from Bacillus thuringiensis, they are popularly called as Bt plants, and they are resistant to many types of insects, especially the caterpillar type of insects, which are mostly the eaters of plant tissues. Another example of this transgenic technology is development of herbicide tolerance. So weeds are everywhere, and most of the time weeds are removed from farmers' field either by manual methods or by spraying weeds with weedicides. But imagine a farm which is actually thousands of acres in land, and which is growing some or useful crop, and it's basically a nightmare to basically manually remove all the weeds from a very big farm. So what people have done, especially in developed countries such as USA, so there are large farms available, and the target crop is made transgenic, where it expresses a particular enzyme which detoxifies the herbicides which can be sprayed. So what happens if, say for example, through some large machines or uh, even airborne methods, helicopters and small aeroplanes, if they spray a very large part of a particular farm or an area with a herbicide, the plants, the crop which is already having a herbicide tolerant gene, they survives, and all other weeds are weeds dies. So that's how herbicide tolerant plants are developed, and most prominent of them are basically the soybeans, which are uh, tolerant or resistant to glyphosate, which is one of the most popular herbicide being used globally. So the transgenic soybean is resistant to glyphosate, and when we spray glyphosate on a field which is growing uh, soybean, the soybean, since it is resistant, it survives, and all other weeds they dies. And basically it basically saves a lot of money in terms of machinery, in terms of fuels which would have been used, in terms of manual labor would have been used uh, to remove those weeds. And if those weeds are not removed, then basically the weeds becomes pest to the crop itself. It basically takes the nutrients from soil. It also takes all the fertilizers which are actually meant for the crop plants, and on the other hand, it becomes host for the pest insects, and then basically it affects the crop in more ways than one. Plants are being make genetically transformed by identifying resistance gene, or popularly called R genes, which protects plant from certain diseases. I'm sure you by now you know some popular plant diseases such as late blight of potato, rusts of wheat, smuts of wheat, and bacterial disease of rice. So most of the time we have two types of uh, plants in a particular crop. Say for example, there are always genotypes which are resistant, and these pathogens cannot affect these fungi, bacteria, viruses. They do not affect some genotypes, and some genotypes which are too sensitive, and they are being affected by these pathogens very easily. So if we can identify the gene responsible for resistance, popularly known as R gene for resistance, and these R gene can be transformed into plant of choice or crop of choice, and then it becomes resistance. Here you see the example of barley plant. So again, rust is a problem with barley, and a gene is identified in another plant, and that R gene is transformed into popular barley cultivar. You can see the transgenic plant. It is completely green in these areas, except the tips, and it looks very healthy. On the other hand, the non-transgenic plant, it is heavily infected by the rust pustules. You can see these brown pustules, and rust fungi basically eats all the food available in the green tissues, and ultimately either plant dies, or even if it doesn't die, it cannot produce enough amount of grains for for any beneficial yield. So most of the time there are R genes, or also called the PR gene, which for pathogen resistance. These are cell wall degrading enzymes such as chitinase. So in case of I'm sure you know the cell wall is made up of cellulose, hemicellulose in case of plant cell, but in case of fungi, the fungi cell walls is made up of chitin, which is again a polymer, and there are genes which code for enzymes called chitinases. So chitinase degrades chitin, same way cellulase degrades cellulose in plant cells. So chitinase degrades chitin in fungal cell, and when it is degraded, the fungal cell basically bursts, and it is no longer viable, and it dies. So transgenic plants are being made in popular cultivars which are overexpressing these PR genes, or pathogen resistance gene. Same thing is done with the resistance gene, which provide resistance towards bacteria as well as viruses and other pathogens. Other use of genetic engineering is making plants which are tolerant for abiotic stresses. So Plants are sessile organisms. They cannot move from one place to another, and if a plant starts growing a particular place, it basically completes its life cycle in that place only. It could be a few weeks, it could be a few months, or it could be many years, but they remain at the same place. And year wise they basically face different stresses. Sometimes there is high light stress, or there is a scarcity of water which causes drought stress, or and these days it is basically an era of global climate change, so temperatures are rising, so plants face rising temperature stress, or also known as heat stress. On the other hand, there can be a low temperature stress also, if temperature goes below the optimum temperature, or they can also face freezing at higher altitudes. So all the times, basically, there, since plants face in any given area, and a perennial plant would face all these types of stresses uh, at a given time point. There can be scarcity of water or induced drought stress, or there can be high temperature causing heat stress. But most of the time, plant survives, although the yield or the productivity of plants affected. But plants do have their survival mechanisms. So scientists have identified genes related for that survival methods, and if these genes are overexpressed, that means the plant will have more adaptations. And it has been found, if these genes are overexpressed, then the plant shows more tolerance or more resistance towards these abiotic stresses. Say for example, heat shock protein genes provides tolerance towards higher temperatures, and many plant species have been made transgenic, which overexpress heat shock protein genes. And it has been found that when the plant is overexpressing heat shock protein genes, then it is more resistant or more tolerant towards rising temperatures, and it gives more yield even if there is a high temperature. Similarly, there are modified sugars which provides drought stress. Say for example, tree halos, fructans, and sugar alcohols such as mannitol. If they are there in plant cell, then plant cell survives the drought conditions much better than the uh, absence of these modified sugars. Also, there is this amino acid proline. High amount of proline, if it's there, then plant cells shows much tolerance to drought in comparison to when proline is in less amount. Another example of genetic engineering based development of useful plant crops is the increase of shelf life. So, as per some studies and estimates, globally around one third of total vegetable and fruit produce degrades because of early ripening, or ripening before it can reach to the consumers. If I give you examples, say for example, apples are in our country. Apples are produced in the hilly areas, mostly in Jammu and Kashmir and Himachal Pradesh. But apples are consumed all over India, and that many times what happens during transportation from the product areas to the consumer areas, the ripening, fast ripening happens, and before it can reach to the consumers, or before it can reach to us, it basically overripe and it cannot be used anymore. Same thing goes for tomatoes or other vegetables. Banana is produced in southern part of India mostly, and when it is transported towards other parts of our country, then during transportation it shows enhanced ripening, and it is many times it happens that it is of no use. So ripening mostly happens by the accumulation of a gaseous hormone called ethylene. Ethylene is a gas, I'm sure you know. So it is called the ripening hormone. When there is more amount of ethylene, then the fruit ripes faster. So basically, it is a mechanism for the maturation of fruits and vegetables. But if it is in more amount, then the ripening happens faster. And sometimes what happens during transportation, that even before it can reach to the target consumer, the over ripening happens, and it is of no use, and it has to be, it's just a wastage. So what people have decided, what scientists have decided, that okay, if the production of ethylene can be shortened or can be the rate can be decreased, that means the ripening would happen, ripening would be delayed, and the resultant fruit or resultant vegetable will have a longer shelf life. As a matter of fact, the first commercialized food crop in the world was a variety of tomato which showed delayed ripening. It was called flavor saver tomato. It was released in USA, and it showed much greater shelf life. As you can see, these are the same age of tomato fruits, tomatoes. This is non-transgenic, which is already ripened, while this is a transgenic tomato showing less ethylene accumulation, and you can see that it can still be kept on shelf for some more days, whether when before actually getting overripe, and also and when it will be of no use. uh, I discussed with you conventional breeding in a little bit extent. So one of the major use, when we say we basically cross different types of plants or contrasting plants. So one of the method of crossing those plants is basically removing male part, or which is a male part from one flower, and using another pollens from some other plant for uh, kind of crossing. One of the method for the removing the anthers of pollen manually is called emasculation, when basically we take a flower and we remove all the anthers so that self-fertilization cannot happen, and we basically introduce pollen from some other plant source to make a controlled crossing in the target flower. As you can see, it's a manual method, and it takes a lot of time as well as lots of efforts to emasculate large number of, or maybe thousands of flowers and thousands of plants in a given experiment for conventional breeding. So scientists have basically, for making hybrids, scientists have designed a method where the it has induced male sterility. So the genes have been identified again from a bacteria called Bacillus amyloliquefaciens, and there is a gene called Barnase. So this Barnase gene codes for a protein, Barnase protein, and this protein is RNA hydrolyzing enzyme. Basically, it degrades RNA. Okay, and when this enzyme is there, so pollen, if it is there in the pollen grains, then it degrades the RNA, and the pollens become non-viable. Even if they are used in fertilization, since they are non-viable pollens, they cannot germinate on stigmas, and they cannot have meaningful fertilization. So they cannot make seeds when the pollens are taken from a plant which is expressing Barnase. At the same time, another system from also identified, which is called bar star. So this protein basically binds to Barnase protein, and it limits activity of Barnase proteins to degrade RNA. So when these two systems are combined, so male sterile plants are made, which used for crop improvement, and it saves lots of time as well as lots of efforts which would have been used in case of induced emasculation for making hybrids in many of the crop plant species. This Barnes bar star system for male sterility has successfully been used for many crops. And one of the most popular example is brassica, the oil crop, mustard. And then there are other uses also, transgenic plants as bioreactors. So I have already discussed the golden rice example. Similarly to golden rice, transgenic wheat and transgenic rice are made, which show higher amount of iron and zinc in seeds. There are transgenic plants made which show higher increased seed proteins, higher lysine content. Lysine is an important amino acid which is many times lacking in vegetarian diet. So high lysine content crops are made, and then crops are being made through transgenic technology, which shows better oil quality. So oil is basically a composition of many types of fats. And it has been found that polyunsaturated fats are good for health rather than polysaturated fats. So, with the help of transgenic technology, brassica and mustards, transgenic mustard has been made, which shows better oil quality. Another exciting research which is going on with the plants is making edible vaccines within the plant food. Say, for example, efforts are successful efforts have been made where cholera vaccine is successfully produced in tomatoes. So, in other words, if we eat tomatoes which has cholera, anti-cholera chemicals inside and anti-cholera compounds inside, then basically we become immune to cholera, and the same efforts are being done for many other diseases. Plastic is getting a big problem these days, and production of biodegradable plastic from bacterial sources is quite an uh, expensive method in comparison to chemical methods of making plastics, which is not biodegradable. And it is a result of three enzymes. All those three enzymes, the genes related to all those three enzymes were actually expressed in Arabidopsis, and results were very encouraging. And these days efforts are being made so that the biodegradable plastic can be produced by large tree species, faster growing tree species such as poplars. Production of crops which are very amenable for production of biofuels. I mean the crops which shows very high growth rate, and they can be easily used for fermentation for the production of biofuels such as ethanol. The efforts are going on for in this direction also. And then metabolic engineering for secondary products is also going on. I discussed with you Agrobacterium tumefaciens, but there is another related to bacteria which is called Agrobacterium rhizogenes. It is also a soil bacteria, and when Agrobacterium rhizogenes infects any plant, so its roots become hairy, and but the biomass increases. So there are much more root in that plant, but that root becomes very hairy. So in laboratory, people, what scientists have designed, that okay, if an explant, any suitable explant for a desired, which produce from a plant from which we need a secondary metabolite, if it is that explant is infected with Agrobacterium rhizogenes, which causes hairy roots, and the same way like Ti plasmid is there in Agrobacterium tumefaciens, Ri plasmid is there in Agrobacterium rhizogenes, and if the tDNA has any useful genes, then the hairy root resultant hairy roots from that explants in liquid culture starts producing the target secondary metabolite, and in this way plants are used, hairy root plants are used as a biofactories for production of important commercial metabolites. And another very classical example of which is very useful for as far as our environment is concerned is the production of colored cotton or colored roses which are not of conventional color. Say for example blue rose, and if we, I mean uh, uh, cotton is used worldwide for production of fabrics, but at the same time those fabrics, to make colors we need to use harmful or pollution dyes which are otherwise pollutes our water source or our soils, and which remains in the environment for many years. So if we can produce some colored cottons, even if they are in some basic colors, say for example red, blue, orange or yellow, then a, a lot of these harmful dyes uh, can be saved from uh, being used in coloring cotton fibers. In our countries also some agriculture universities are doing efforts. So basically taking genes which provide colors to certain flowers. So for example, there are genes which provide color to different type of roses or petunias or any other uh, another popular flower. If we can identify those genes, and those genes can be overexpressed in cotton balls, then that cotton will have some color. So as a proof of concept, scientists have already made blue color cotton in laboratory. One one issue, or one of the section, deals with the biosafety issues in plant genetic engineering. Since it is an induced method, it's a lab-based method. So like any other industrial method or chemical method or any other improvement method, the effects of plant genetic engineering needs to be studied in detail before the product is released for commercialization or for common use. These issues are mostly related into 3-4 categories, such as they need to be tested for any adverse effect on humans, if they need to be consumed, or in another animals, which can use plants for various things. Most common is for the foods. The genetically modified plants are also needs to be tested for their surroundings, for the nearby biodiversity. They should not have any bad effect on nearby plants. Same way, they should not have effect on useful insects. Say, for example, honeybees. They are very useful insect. So if cotton plants are Bt cotton is there, so the delta endotoxin, they should not have a harmful effect on non-targeted insects such as butterflies, and basically they are Bt cotton is very safe. But the same way, if any other gene or any other protein is tested for insecticide properties, it needs to be tested for non-targeted beneficial insects as well. It should not kill the beneficial or the other insects which are there in the same environment. Also, it needs to be tested if the pollens of transgenic crops basically can fertilize other crops or the other plants. It is not desirable if it happens. And then if it is a food crop, then it needs to be tested for uh, any undesirable effect on animals or on humans, or if there is any test related to allergic or any other undesirable effects needs to be done in a controlled way before it can be released for general use. Same way, it needs to be tested that the transgene should not be horizontally transformed to the non-transgenic plants or some other plants through the action of air or through the action of insects or any other action. It should not be transferable from one plant species to another plant species which is growing nearby. And last but not least, the overexpression or the effect of gene which has been used in transformation needs to be studied whether it is not causing any evolutionary changes or induced mutations in the target plant. Even after all these things are done, it's been almost now 25-26 years when first genetically modified crop was released. I think it was in 1986-87 when the first time this flavor saver tomato was released. Since then, because they are so useful, the use or the adoption of GM crops has seen kind of an exponential increase. So starting in 1996, not 1986, as I mentioned earlier. So starting from 1996, when only 1.7 million hectares area was under GM crops, in 2016, in a span of 20 years, you can see that from merely 1.7 million hectares, the area increased to 185 million hectares, more than 100 times increase within a span of 20 years. And there are now around 26-27 countries which are growing different type of biotech crops. India is also there in this list, and we grow cotton, Bt cotton, as a GM crop in our country. The major biotech crops or major GM crops are soybean, maize, cotton, and canola. So the largest or largest crop which is being grown as a GM crop is herbicide tolerant soybean, as I just mentioned in one of the slide. And mostly it is grown in Brazil and in USA because they have very big farms. So what they grow soybean as a feed for their meat production thing. And the soybean is what they grow is genetically modified for herbicide tolerance, glyphosate tolerance, and then whenever they spray glyphosate, the soybean remains alive and all other weeds dies. The second biotech crop is staked traits maize, which is basically have many traits, and those traits are provided by Transgenic. And third one is Bt cotton, the cotton which is resistant to cotton bollworm. Other crops which are being grown worldwide and food crops are brinjals, and then these are squashes, potatoes, apples, and rice and wheat as well, and papaya and banana. The golden banana is there now, golden maize is also there now with increased amount of beta carotene, same way as golden rice is there. So after going through these videos again, I put certain questions which you should be able to answer, and there are many other questions which can be answered by seeing this video. And some of the questions are: what is genetic engineering of plants? What are the major steps in plant genetic engineering? What is agrobacterium and what is dihyplasmid in agrobacterium? What are the methods for direct DNA transfer, or one method is microprojectile and gene gun. Others are electroporation and microinjection. What is golden rice, or golden corn, or golden banana, and which was the first ever genetically modified food crop. And I just discussed which is the genetically modified crop which is grown in India, and what are the other major crops which are already genetically modified in the world. You can refer to that table as well as to the figures in the previous slide. And with this, I would like to end this video with this figure, which is the blue rose, which was produced by a Japanese company called Suntory Limited. So interestingly, the rose doesn't occur in actually pure blue color. So Suntory Limited, they took this blue color gene from some other flower, and then this gene is introduced through genetic modification in roses, and they basically got a gradient of blue color. And some of the resultant plants, they have kind of true blue color. This one is violet, but there are pictures available which shows kind of true blue color in transgenic rose. So with this, I would like to end this video with a note that genetic engineering offers us too many uh, too many methods by which we can achieve unconventional things in plants. Say, for example, if it is related to better food crops, high nutrition, or better resistance towards abiotic stresses, or better resistance towards insects and pests, which would ultimately increase farmers' yield by reducing the inputs as well as by saving crops from pests and other stresses. I'm sure these videos would benefit you in understanding the concepts of plant cell culture and its application, which is related to chapter 6.2. Thank you very much.